Humans and animals undergo remarkable journeys of growth, starting from conception and progressing through infancy, childhood, and into adulthood. Their development shapes their physical, cognitive, and emotional selves. Similarly, animals navigate their own paths of growth and maturation, from a vulnerable stage in infancy to strength in adulthood, adapting to their environments along the way. However, amidst these visible growth processes, have you ever wondered how microbes grow? To understand this, we will explore the phases of bacterial growth, which provide insights into the dynamic processes of microbial proliferation.
Let's start with the lag phase. Lag phase is an adaptation period where bacteria, whether slow or fast-growing, undergo adjustments to their new conditions. However, the duration and characteristics of the lag phase differ between slow and fast-growing bacterial strains.
During the lag phase, slow-growing bacteria such as Mycobacterium tuberculosis, the causative agent of tuberculosis, undergo an extended period of adjustment. It requires ample time to synthesize RNA, enzymes, and essential metabolites necessary for survival and replication. Conversely, fast-growing bacteria like Escherichia coli experience a shorter lag phase due to their rapid adaptability to environmental changes. E. coli swiftly adjusts to nutrient availability, enabling a quick transition to exponential growth. Although fast-growing bacteria invest less time in repair processes during the lag phase, their adaptability ensures efficient utilization of available resources.
Once bacteria accumulate necessary components for growth, they enter the exponential or log phase.
In this phase, cells undergo rapid division resulting in predictable population doublings. Conditions favoring bacterial growth lead to exponential increases in population size. Cells in the exponential phase of growth are the healthiest and most uniform, which explains why most experiments utilize cells from this phase. Due to the predictability of growth in this phase, it can be used to mathematically calculate the time it takes for the bacterial population to double in number, known as the generation time. The generation time is a measure of how quickly the population is expanding and can vary depending on factors such as the specific microorganism, the environmental conditions, and the availability of nutrients.
As bacterial growth progresses, limitations such as nutrient depletion or waste accumulation lead to the stationary phase. In this phase, cell growth ceases and the number of new cells equals the number dying off.
Physiologically, cells undergo changes to adapt to starvation conditions, becoming smaller and more spherical with altered plasma membrane properties and condensed DNA, similar to what is observed in E. coli. These adaptations promote survival until more favorable conditions arise. During the stationary phase, bacteria such as Bacillus subtilis exhibit the production of secondary metabolites, including antibiotics, and initiate sporulation in cells capable of forming endospores. This phase reflects strategies used by cells in low-nutrient environments and suggests that in nature, cells often experience prolonged periods of nutrient scarcity interspersed with brief periods of growth.
In the death or decline phase, deteriorating culture conditions lead to a decrease in viable cells. Despite this decline, some cells enter a viable but non-culturable state, exhibiting low metabolism until conditions improve.
Examples of bacteria that can enter the VBNC state during the death phase include Vibrio cholerae, Escherichia coli, Salmonella enterica, and Mycobacterium tuberculosis. Complete cell death is rare due to mutation and adaptation, often resulting in a tailing effect where small population persists. Additionally, the death of cells benefits surviving cells by releasing nutrients into the environment.
It is important to learn about the stages of microbial growth because it is essential in a wide range of applications, from healthcare and food safety to environmental management, contributing to the improvement of these sectors. I am at the Cancabatu Bay, a coastal body of water located here in Tacloban City. This bay is a crucial water source for the residents, supporting fishing, recreational activities, and other several uses. Part of the environmental management is ensuring the safety and quality of the water. So how is it done? A very useful parameter in assessing water quality is to determine how much microbial growth is in the water. We will be talking about several ways as to how microbial growth is measured. The growth of microbial populations can be measured through direct and indirect means. Direct methods of measuring microbial growth involve directly counting individual microbial cells or colonies to assess their proliferation.
The first one is the direct microscopic cell count.
In this method, a precise volume of bacterial suspension is placed on a microscope slide within a defined area. A specially designed slide called Petroff-Hauser cell counter is used. A 0.01 milliliter sample is spread over a marked square centimeter, stained for visibility, and viewed under an oil immersion objective lens. The viewing field's area is determined, and bacteria are counted across multiple fields to calculate the average per field. From this, the bacteria per square centimeter and per milliliter of the suspension are derived.
The next one is the plate count, which is the most frequently used method. Plate counts are typically reported as colony-forming units or CFU. Microbiologists prefer plates with 30 to 300 colonies. To achieve colonies within this range, the original inoculum is diluted several times in a process called serial dilution.
A plate count is done by either the pour plate method or the spread plate method. In the pour plate method, either 1 milliliter or 0.1 milliliter of diluted bacterial suspension is introduced into a petri dish. Liquid nutrient agar is poured over the sample and gently mixed to ensure even distribution. After solidification, the plate is incubated. However, this technique may damage heat-sensitive microorganisms and will therefore be unable to form colonies. Alternatively, the spread plate method is often preferred. Here, a 0.1 milliliter inoculum is spread uniformly onto the surface of a pre-poured agar medium using a sterilized rod, ensuring all colonies remain on the surface and preventing contact with melted agar.
When the quantity of bacteria is very small, as in lakes or relatively pure streams, bacteria can be counted by membrane filtration technique.
Water samples are filtered through a membrane with pores too small for bacteria to pass, allowing the bacteria to be retained on the filter surface. The filter is then transferred to a petri dish with nutrient medium, fostering colony growth.
This technique is commonly employed for detecting and counting coliform bacteria, which are indicators of fecal contamination of food or water. Another method for determining the number of bacteria in a sample is a most probable number method. It relies on the fact that the greater the number of bacteria in a sample, the greater the dilution required to reduce the density until no bacteria grow in the tubes of a dilution series.
In this method, an MPN table is used to calculate the microbial numbers that are likely to lead to such a result. The number of positive tubes is recorded for each set. In this example, the number is 5, 3, and 1. If we look up this combination in an MPN table, we find that the MPN index per 100 ml is 110. This means that 95% of the water samples that give this result contain 34 to 250 bacteria, with 110 being the most probable number.
The method is valuable for microbes that don't grow on solid media such as chemoautotrophic nitrifying bacteria, or when liquid media are used for identification of microbes such as coliform bacteria in water testing.
However, it is not always necessary to count microbial cells to estimate their numbers. Indirect methods, which measure parameters associated with microbial growth without directly counting cells, are also used. For some types of experimental work, estimating turbidity is a practical way of monitoring bacterial growth. The instrument used to measure turbidity is a spectrophotometer. However, turbidity is not useful for detecting low levels of contamination. About 10 million to 100 million cells per milliliter are needed to make a suspension turbid enough to be read on the spectrophotometer.
For filamentous bacteria and molds, the usual measuring methods are inadequate. A better approach in measuring the growth of filamentous organisms is by measuring the dry weight.
In this procedure, the fungus is removed from the growth medium, filtered to remove extraneous material, dried in a desiccator, and then weighed. The same procedure is followed for bacteria.
Now that we have learned the stages of microbial growth and the different methods in measuring microbial populations, we can effectively monitor and manage microbial growth in various environments, ensuring safety and quality in industries such as food production, healthcare, and environmental science.